this side seems to have a lot of life on it.
A little bit of stuff on there.
Teeming with life, yeah. What's the top look like? Nice, Jake. That's a cool shot.
Yeah.
Yeah, this is it would be really neat if we did like a mosaic of this rock.
Photogrammetry.
It's one of those really great mosaic targets.
Okay.
No, we don't have to do that. It takes a while. But this is one of these features, right, that shows you three-dimensionality.
Mm-hmm.
That isn't always clear from looking at a 2D video.
There's so much here.
So cool.
Is this a good EDNA candidate location?
But yeah, wow. Yeah, there's a lot of stuff on here.
This is a forest.
Like
yeah.
every available.
I really wonder why they picked this rock.
Seems like it's just there was the walls before, but this is a pretty
Yeah, this is
less
just like a
steep
little
slope, and then
yeah.
there's just this one erratic.
Yeah.
On some of the last watch, they were coming across these boulders as well.
Yeah.
Just like covered.
Maybe that's the preference.
Yeah, this is where the current brings the food.
Very cool.
Come up, Jake.
Coming up.
Yeah, Jake.
I was in the process.
We like your shot.
Yeah.
Really nice shot.
Hey, Steve, does this qualify as a garden?
I would say so, yeah.
All right, probably have to get on track.
Certainly
here
has the
again.
density.
Say it's a raised bed.
Very, very cool, though.
Oh, the jokes are so good. All right.
All right, we're going to get check in again soon, but one last look at the rock, I suppose. Beautiful colony.
Yeah. Wow.
And it's very clear that the current must come from this direction because like view in Argus's cam, there's nothing on the other side.
Mm-hmm.
Yeah.
Yep.
All right.
All right, what is our current heading? Okay. Yep.
Make up some ground here.
Well, I guess we added some support to the theory that it's not entirely boring up on top of the flat part up here.
Yeah, we are getting up to the top, aren't we?
Yeah, we'll see if we can find the kind of local high here. We might end up. It's hard to say exactly. Just we have a course resolution, but
yeah.
So we may end up on the northwestern flank of this, and we can kind of figure out where we want to go from there.
Yeah. Yeah, so any thoughts, team, about which direction we might want to go next? Do we want to start heading kind of southwest along this? Do we want to try to go back down to any particular depth to the southeast?
Whatever you guys think back there.
Any thoughts, Lindsay, Steve? Folks at home?
Folks at home. Folks at home.
Yeah.
They want to go right.
Yeah, they really want to go right, but we're
Yeah.
not going right.
We came from the right.
You know, we have a lot of runway here.
Yeah.
Even if we finish the dive and get to the end point, we can still keep going. It goes up for another several hundred meters.
Yeah.
So I am of the opinion that, you know, we know that the steeps are kind of, you know, has really nice corals scattered. I'm of the opinion that I might want to look for some distance on the top of the tongue here.
Okay.
And just see what it's like for a little bit, see what the variation in the community is like over, you know, 100, 200 meters, something like that.
Okay.
But
so if I'm grasping that right, you want to go southwest along the top?
yeah, that's what I would say. And if it really is just ultra dull, we can always drop back down, right?
Right. Not just dull, but ultra dull.
So let's see what's happening here. It looks
It's
like
like pretty flat.
Yeah, I'd like to look at a lot of different types of
Yeah.
terrain.
Yeah.
You know, ridges are nice, but you'll sometimes have totally different community structure on slopes of 60 degrees plus compared to 30 to 60 degrees, and it's important to take a look at all of those.
All right, let's go 220 then. Are we in agreement back there?
Yeah, that looks
Okay.
good where you've got the cursor.
Nice.
Yeah, so I'll Argus is still gonna swing a bit this way, and then, but I'll call the move in now, and we'll kind of loop around. 220.
220?
Raj.
Bridge nav.
Do you want me on 220, Jess?
Step 100 meters, 100 meters, bearing 220.
We can also just lateral. Oh, sorry.
Lateral.
Thank you. Yeah, I think, Jake, you'll probably still have some 315.
Okay.
And then, I don't know how Jess wants you to curve it around. Or
Yep. Okay.
Gets better images. So yeah, if you're following along at home, you can see in the lower right quadrant exactly what we're describing here. You can see that the green ship, which is tethered directly to Argus. Argus is the
little square with the blue line is the track that Argus has made, and the little square here, I don't even know if you could resolve it at home, is Argus, and right next to it is Hercules. Hercules has a red trail of where we've been, and it's kind of giant cursor pointing to where it's heading.
cool. And can you point Remy in the direction that we're going to be moving
Yeah,
at?
so the ship still, Argus has a little bit of movement for the ship, so we'll catch up, and but I just called in a move to go bearing 220, which is approximately southwest. We're looking at these contours here indicate that we'll be traversing along the top of this larger ridge feature, which may or may not be flat as we're seeing it, or it might have features that we couldn't resolve in multi-view mapping, like these are boulders sticking out, or maybe ridge features of the base rock.
Jeff, you want to do a partial there?
So one of those,
Sculpins.
I'm going to get it, Tronoclops. Was that right?
Tronoclops?
Yeah,
Tronoclops,
on
yeah.
crops.
That unbranched white stalk at 9 o'clock is very interesting. Can we focus on that a little bit closer?
This right here?
Yeah, no, up in the upper left, you see there's unbranched white.
Oh, next to the mushroom coral?
Next to, yep.
Sure.
That looks similar to that thing we were looking at earlier, right? Where
Yeah,
it
I'm
ended up being,
wondering if it's it might be a small paragorgia too, like a Cipogorgia that just is very young, and it hasn't branched out yet.
Or the stolonifera.
Yeah,
Either.
this one has a little bit more opacity to the axis, so it makes me think that it's actually producing that skeleton.
I'll just rest here, I think, and then we can zoom in on whatever we like. Never mind.
Well, if I had I wish I had another rack of Niskins, because I would probably be popping Niskins all the time. I didn't only have 6.
This is making me wonder if that last one was.
I really want to sample this thing.
This
Is there
white.
any
We can, if you want to
do you have
stop.
time?
Yeah.
If
I'll
it's
hold here. I think we should.
a burden, we can
I think it'll be all right.
for another one.
Bridge nav.
Do you want, is it with
Hold
the
position
slurp
here.
or in the bio box?
Thank you.
It doesn't seem like it's on a rock we can pick up.
I think Argus will pass over you, but you'll still have enough tether on the other side.
All right. Yeah, we've got.
So the slurp is not
40, 30.
an option,
Yeah,
right?
I think Argus will end up here.
Right now for this thing.
Okay. Thanks, Ray.
What's your professional opinions?
Sorry, what was that, Steve?
Slurp, is it functional?
We can take a hair trim off of it, and then, well, we're kind of occluded here, aren't we? Let me find another angle. We can take a hair trim and try it in the slurp, and if that doesn't work, we'll put it in a box.
I know it's in a really awkward place, but do you think you can get a toe in there to sit down in front of that?
A toe in front of it?
Yeah, like so we can zoom in close before we sample it?
Yeah, it looked like it was hidden if I tried that ledge, but I can try here, and then we'll see if this works.
Yeah, that pink coral to the lower left is going to be pretty fragile.
Roger.
Won't be flexible if you...
Jake, you want to give me some delta here?
Yep.
I'm down.
Huh. You're in a fun location.
You know, I think we can try and do the slurp. I'm talking to the back row here, but we can try and do the slurp and see how it goes. But
snip and slurp or just
snip and slurp? Yeah.
Oh yeah.
I would also consider putting it in a compartment with the rocks, if on the starboard side, the angular rocks. That would be okay for me too.
Tough sample spot.
Since I'm pretty sure I can tell this coral apart from rocks.
Let's try over here. Yeah, what about
Jake,
the other side? Turn and face me here, and then also if you go to 10 on your delta, if the sonar permits.
My sub-bottom,
Sub-bottom's at 10.
It looks
Oh, okay, never
I can
mind.
come down like another meter or two.
Roger.
Oh, too far, too far.
It's a very interesting position, this one.
Might be at the edge here.
Let's do right here. Randy, you want to do me a favor?
Yeah. Stick lock.
Stick lock.
How much time do you think we have, Rennie?
Go ahead and push on in there a bit, please.
Or should we?
I'm trying to figure out how much Argus is moving or not. We can have as much time as want. I can just try to bump the ship towards Hercules.
Okay.
I'm just trying to figure out what we did a little bit of a curve move, and I don't know where we're going to end up.
All right, Steve. So this is a pretty bad shot for you. But I think
if I head north, we'll be okay.
You push in all the way that you can go. Images first. Yeah, I really would like a piece of this.
Sure.
It's I don't even know Now we've got 2 colonies. They could be stoloniferans, but it might be something else. I'm very interested. So any of that we can get would be a pretty big deal.
Sure thing. All right, full wide there, please.
Can you range in on the sub bottom? Jake?
Yeah.
So we can get it better. Sorry.
10 meters.
If it does go north, I think there's a slight rise, so we'll have to go up a bit.
I'll come up.
At that point, you'll be above Herc anyway. So.
Yeah. All right, we're steady, kind of. That's not a very good parking spot, is it? Let's increase the joy gain, maybe that'll help us here. All right, Jeff, you want to push in there slightly, please? That's great. All right, you want to come a little wide so I can see the jaws? See. All right, you want to push in there a little bit more, please? All right, got one shot at this dude. Swing and a miss. Here, Annie, can you get this glasses case out of my way here?
Yes.
Thanks. Perfect.
Nice.
Sorry,
Very
I
cool.
apologize.
That's good.
What's your professional opinion? You think you're gonna get that in the slurp jar, or to be on the safe side?
To be on the safe side, I would probably put it in a box.
Okay, let's go in the box.
Especially because the sample, we probably won't get 2 of it.
We can get it in the hose. We know that. Yeah.
You can come full wide there, please, Jeff.
Fish it out on deck.
Yeah.
The hose is a
Yeah.
place where samples go to disappear forever.
Yeah.
Yeah. What would you like to do with this guy?
I mean, we can also use those front bio boxes if you want, because
No.
most of what we put in there is pretty small.
We can double it up.
Okay.
Yeah, we can double it up. Yeah, it's so distinct that
Yeah.
we'll be able to tell it apart. So let's just dump it in one of the outboard starboard boxes,
Okay, sure thing.
so we don't have to deal with the floatiness
Yeah,
issue.
I like the idea. Before we kick it out there, Jake, let me just get resituated somewhere else so that
Alright.
we don't
Sounds
knock
good.
all of our samples out. Very cool.
Let's get a target there while we're at it.
Let's go right up here. Lots of bravos and appreciations for that snip.
Yeah, that's
Jess.
pretty incredible. With the glasses case in the way too.
With the glasses. Yeah.
Yeah. Not only was the boat rocking, but Herc was rocking back and
Herc was rocking, boat's rocking.
forth.
And my glasses are in the way.
Right, essentially there was a brick under the brake pedal.
So really it was Rennie that saved the day here.
Okay, let's not celebrate
Until we
someone
get
who
in
does
the
not
bin.
need it.
Roger. All right.
That was all you.
Sit down here. All right. Did you want to screen grab of this, or are you good with just stowing it?
We can stow it. I got some good images during the collection.
Roger that. Jake, you want to follow me around in bubble?
Yep.
Thank you.
You don't think it'll be too floaty in the outboard big box?
I don't think so. I think it has some weight to it.
Okay.
Which one are we going to there again, Steve? F?
E or F, it doesn't matter, yeah.
Okay.
Whichever is more secure.
Roger.
There's a bunch of rocks in there already, so I think it won't get crushed since there's already rocks in there.
Sure thing.
This will float to the bottom and just kind of sit there.
I heard that there was a floaty in F, right? So
In A, so just open it halfway. Sure.
Roger.
Oh, you know what? I just had a great idea. We can use those unused quivers as
Oh, yeah.
as if you have a stopper, we can
Go ahead,
make
Jake.
them
All right, come
sample
halfway.
quivers.
Halfway. Right.
Yeah, I mean, we've got 3, and I don't know that. Oh, something just.
Oh, little.
That's good there.
That's okay. I don't think it's the sample. That's too small.
Yeah, okay. But yeah, you're right. We could use the sediment quivers.
Yeah.
Push cores.
Okay, we're going to go for F here, Steve, okay?
F, got it. Okay.
F for Frank. Just shove our claws down in here so we don't lose the sample.
Looks like it
All right.
slid out.
Looks like it fell. Yeah.
Did fall, yeah.
All right. Claws of Doom. Cool.
Definitely went in there.
Yeah, it went in there.
Great. Great. Awesome.
White unknown octocoral.
What was the sample number?
036. Yeah, that'll be very interesting to look at. Can't wait to take a peek at that one later.
Cool. Thanks, guys. All right, now we can go back on track.
Yeah. Ready to move onward, or you need some more imaging back at that boulder?
No.
Yeah, no, I think we can keep going.
All right. 100 meters at 220.
220.
100 meters, 220, Raj.
Bridge nav. One step, 100 meters, 100 meters, bearing 220. Thank you.
Jake, if you want to go back to 310, I'll just lateral and we can see the slope.
Back to,
Yeah, like 310.
yeah, that's, yep. Headed.
Come up a bit on the delta there. Steve, are you able to describe any of the particular features that were striking you as unusual or particularly exciting about that sample?
Yeah, it's so the polyps are reminiscent of a stoloniferan, but it's the second time we've seen it on a stalk of some type, which makes me think that maybe it might be some other group that I'm not familiar with or haven't seen very often, because it could be that it produces that stalk. Stoloniferans typically don't produce their own stalks. They typically run along rocks connected by tissue. The polyps were extremely large for the size of the skeleton that it was on, and the sclerites were very obvious through the body wall of each polyp. So you can actually make out with the video we had the small sclerites, calcium carbonate structures in the polyp.
Through those kind of white lines.
That's typical of
Roger
soft
that.
corals, like true soft corals.
Are there examples of parasitic corals?
Yes. I've seen a couple of different types of stoloniferans, for example, that will parasitize a sponge and grow from within and through the sponge. So it's basically using the sponge's body as their substrate.
Definitely more sediment debris in the water here. There's a stalked sponge off to the left and one right ahead of us. Or no, that's maybe not a sponge, but
Yeah, there's still a fair bit of corals up here.
yeah. There's definitely
Sponges.
some current and sediment moving through here.
Yeah. Maybe the current's on the other side of this.
Yeah, maybe it's coming from the west instead of from the east.
A lot of the times these the smaller corals that don't produce large skeletons are often poorly represented in our knowledge about corals. Just because they are so small, it's very difficult to get a very good specimen. Whereas if you were to, these larger charismatic coral fauna, like the Iridogorgia here, some of the large bamboo corals, those are probably sampled more frequently than some of these smaller ones. But all biodiversity is important. All biodiversity is information. It's genetic resources. It's so it's important to get a good picture of everything that's living here, not just the big things.
Looks like there might be in one of those glass sponges in that coral. Well, it's kind of hard to tell from this distance.
Folks wondering what the V is on their screen, but what we're actually seeing, I think, is that
The lasers.
laser showing up with the higher sediment in the water.
Yeah, exactly. There's more particles in the water that are catching the two parallel lasers that are 10 centimeters apart.
Right. Oh, you're good there, Jake, actually.
Okay.
All right, let me redo this. Sorry, guys.
You're at the end of your leash.
Sure. Just gonna straighten myself out here a little bit and get out in front. Sorry for the blue water for a minute.
It's okay. Do what you got to do.
Yeah, while we're in the blue water, folks are kind of wondering if this looks like they've seen a few of our dives now and noting that there seems to be a higher concentration of life on this dive than the previous dive. Was that expected? I believe it was. Do you want to describe kind of why there might be some differences there, Beth?
Yeah, sure. So today's dive and yesterday's dive are both on Haheo Seamount, which is, oh, hold on one second, pull up my map. We're kind of due south of Kamole or Leisan Island, and that is an area, previous work in the monument has indicated that there's relatively high coral diversity in this region. And so a question of our diving program is to see if that extends to these deeper seamounts and especially at these deeper depths. So yesterday's dive started at the southern end of the seamount, working our way from south to north. Today's dive, we're on the north end of the seamount, working our way from north to south. So we're potentially closer to that known coral diversity, being on the north side. And current probably has a lot to do with the spread of those organisms. And so we're still trying to understand which way the current moves predominantly around the seamount. We're learning that as we're doing these dives. So both of those factors may be part of what we're seeing. We're also at, I believe, a shallower depth than we were on yesterday's dive. Yesterday we started pretty deep at 3,000 meters. And I'm not sure what our final shallowest depth was, but I think it was shallower than where we are now. So all of those factors combined are probably playing a role in helping us, or in supporting a larger animal diversity and abundance here than what we saw in yesterday's dive.
Thank you. Wonderful overview.
Yeah, great question. And anybody watching online, please feel free to use that message function to send us questions or your thoughts about what we're seeing. We know there's experts all over the world in these animals that we're seeing. So appreciate your feedback.
Curious if you have any thoughts, Kalame, who, as you're watching, and
my appreciation for all of you guys' knowledge and experiences grows by the minute. But otherwise, learning plenty.
I'm curious, Kalame, when you were being invited to participate in this, did you have an idea of kind of what it would be like to see the seafloor and experience this? And is it kind of matching your expectations? Is it wildly different?
I didn't really have any expectations.
Okay.
I mean, I think that the story that we've been sharing is that like, sorry. My expectations were that, you know, we're just gonna be here for the Olelo Hawaii liaison position, and so I was just really excited to be able to interact with our communities from the ship and stuff. And I think that anything else is quote unquote extra credit.
Yeah, great.
So it's been a very eye-opening experience in many ways.
Yeah.
I definitely appreciate the various ways in which knowledge is gathered and the way that it's like, I don't know, just expressed and stuff like that. It's just a big culture shock.
Yeah.
But definitely important. And so I hope that our viewers out there, whether you're, I mean, specifically in Hawaii too, like just because I'm from Hawaii, so there's a bias, right? But I hope that you're watching this and just understanding that you can definitely be here in any of these positions too. So
yeah,
in the beginning, right, I was just kind of like, oh, talk story. That's kind of my thing. It's easier that way. But as I see it now, I see the importance of being able to come, for Nautilus to come to these communities.
Yeah.
to be here and to create pathways for us to be connected in this way, and hopefully reciprocal avenues to share the knowledge that we gather here, the research, and all that kind of stuff. I'm sure there's a lot of that going on behind the scenes from what I don't see, but otherwise, yeah.
Very cool.
Happy to have you out here.
Yeah.
Thanks for having us, gang.
Jess, is it possible to pivot to the right so I can look a little bit more at this sediment material that we're crossing over? Oh, I don't think you're on SPL.
Yeah, sure thing.
Yeah, you should have a little bit of time here.
I'm curious if this might be something I could get a sediment core in, but I kind of want to see what the surface looks like. If it's like
Sure.
it's like these
You
tiny
can just
little
land
nodules.
here.
It's gravelly.
Yeah, I don't know. It doesn't look like it'll be very good in the quiver, or in the core rather, but
Yeah,
It would be worth a try.
it would be worth a try.
Do we have the scoop on board?
we have a scoop. Yeah, that might be more.
Argus is directly above Hercules. The ship has finished its move, but I expect it'll still go southwest, so we'll have to backtrack if we do indeed want this. So let me know once we poke at it.
Yeah, if we could just get a partial zoom.
Go ahead there,
See
Jeff.
kind of what this texture looks like a little bit more.
Yeah, you're right. It does look like.
Yeah, lots of little nodules. And it looks like the sediment kind of goes a little bit farther to our, the right side of Herc. So maybe there's some spots to try poking around and see if we can get something.
Sure.
The scoop will definitely get us the nodules, but it won't get us the sediment, and that's kind of an additional question we have. So yeah, if we could give it a try with one of the push cores, just see.
Okay, I'm going to backtrack a little bit.
Okay, thank you, Rennie.
Yeah, we could also. Do you think we'll be okay here, Rennie? Or we can also, I think, pop up over right there. What do you think?
Yeah, we're looking at going a little farther up to see if that's the same type, Beth, up there in the Argus cam.
If it's the same what?
The same type of material as
Okay.
we're looking at here.
That's off to our left?
Yeah.
Yeah, up to the upper
I
left.
just dropped a target in case we have to come back to this, just to see.
Okay.
Oh wait, we won't get tugged on our way.
Yeah.
But we are on the way back to amend that.
Let's see. It doesn't look the similar.
No.
No.
No, yeah. Okay. Go back down.
It's a bit upslope, so we might have to pause until we
Yeah.
get back into position.
Yeah,
Yeah.
agreed.
Yeah.
Beth, we're going to wait for the ship to kind of settle
Yep,
out.
that's
and
fine.
then we'll go back down.
Okay.
Are you going to be moving far from where you are now?
Ideally not. Probably 20 meters or so at the most. Coming up.
Why is that? Do you need something?
No, I was just curious. I saw something, but it can wait. Or pass. It's not urgent.
We'll be going back down there though if we want
Okay.
the sample.
Yeah. Keep an eye out for it.
Yeah, it's hard to tell now where Argus is going to end up because once we got to the top of that ridge, that current actually ended up really blowing it to the southeast.
Okay.
So it's settling into its new position that it wants to live in this current. But look at those things.
More corals.
This is just a preview. We got a little sneak peek.
Little sneak peek.
And we retreat.
Yeah, Beth, I am curious though. It doesn't seem like it will make an intact core. And I would imagine that there's actually a pretty hard substrate underneath, like a pretty superficial layer of softer sediment. So I'm not sure if we're gonna get a good core. If not, are you guys comfortable with the scoop?
Yeah, so the scoop won't get us any sediment. And the question to you is, could we use the push core and kind of move it horizontally instead of vertically to try to capture stuff and then get it in the quiver?
Yeah, so the only issue with that is the actual process of getting it back into the quiver. When we flip it vertically, it's all just going to fall out pretty quickly.
Yeah.
Especially, yeah, since it's not, it won't be consolidated, I don't think.
Right.
Yeah.
You won't be able to get that vacuum if you don't put
Oh
it
yeah,
into
the nodules, if they fall in, then they,
Yeah, it's true too.
Do you have a fine mesh on any of the slurps?
No.
No, nothing that would stop that. It looks pretty thin sediment, doesn't it?
Yeah,
Anyway.
it's okay.
Well, we can try all of these.
Especially since we're moving kind of far away at this point.
Beth, what are these nodules you're speaking of?
Excuse me?
What are nodules?
The manganese nodules. So they are a type of mineral formation that generally is seeded by something small, like a sand grain or like a clump of sand, shark's tooth, all kinds of things that slowly over many tens of thousands, hundreds of thousands of years, metals precipitate out of the water onto the surface of these and form kind of layer upon layer upon layer of metal oxides that also co-precipitate many other types of metals. So they're often just referred to as a manganese nodule or a ferromanganese nodule because manganese and iron are the dominant elements, metals. And they range in size from, very small, centimeter size, half an inch or so, up to, kind of the size of a big potato. Just kind of depends on where you are. So we've seen on a couple of different spots on this seamount areas where there seems to be kind of a depression, maybe a little flatter area, and you have these kind of pebbly sized manganese nodules.
Very cool, thank you. I think that was a little different than some of our viewers were expecting, so that's super helpful.
Yeah, so unlike most of the rocks we're seeing on the seafloor here, which were formed from, cooling of superheated molten rock, the way that manganese nodules formed is quite different. And you can have these manganese, the same ferromanganese crust that form nodules also form on these
on
basalts.
larger surfaces.
Yeah.
Yeah.
So some of the rocks that we've collected have an inner core of a original basalt, and then they have these crusts that are formed on the outside as well. Coming back to the conversation with Jess. in terms of what we might want to try. So I don't know. I have kind of lost our situational awareness of where we are now versus where we were with that idea of doing sediment. But I don't know if it makes sense to go back at this point or if we just kind of.
Yeah, so we're about halfway back.
Okay.
We're swinging back. Yeah, I mean, it'd be very interesting. We can also just probe the sand quickly and or probe the sediment quickly and see how consolidated it is.
Okay.
So we do want a good plug in order to get a good core.
Sure.
Yeah, so we can probe to see what the depth is and how consolidated it is. It is consolidated and it's somewhat deeper than we can go ahead and take a core, and that'd be quite surprising. If not, we can also just try taking a scoop if you guys have an available bin or want to cross it into a bin with more biology. I'm not sure.
Yeah, I don't know. Does any of the sediment make it, not in the starboard usually, but in the forward bio, does that sediment make it to the surface? We try to fill up a bin.
Yeah, we can.
Are you just interested in the sediment, or were you actually interested in the layers?
Yeah, ideally we want. Ideally, I would like a consolidated sediment, just so that it's not completely disturbed and mostly seawater.
Yeah. Go for it. We'll see if it pours.
Yeah, and where we were, like where we sat down, kind of was like. I'd been looking at it for a little bit, and then I'm like, okay, maybe we should just check it out. So maybe there's some spots.
Yeah.
Like it seemed like a relatively extensive feature, so.
Yeah, we're on our way back now.
We can just drive over it and see like where it looks like it might be thickest.
Sure thing, yeah. Let's have a look.
Roughly where that red cross is.
You rock, thank you. Coming down a little bit.
See that thing coming into the laser? Is that stock?
Looks like a stalk from a sponge.
Yeah, that's the thing I was wondering if we could take a quick glance at as we go by. Zoom in on it. I didn't think we'd see this again,
go ahead.
actually. We kind of ended off.
That's one more of those.
It's the thing we just sampled.
Wow, and it's so much easier location. Wow.
And it's attached to a rock.
It's attached to a rock.
Is it? Those polyps look so much smaller.
Yeah.
Yeah, no, it's the same. So I'm thinking that this actually is a stoloniferan now, and then it just is on different substrates.
Yeah.
And this looks like a sponge stock, like a caulophacus stock.
Yeah.
So what's happening here is this coral is not producing this stock. That's what I think we've established now after 3 observations. It's actually it's opportunist. But it's good. Then now we know that it's definitely a stoloniferan, and that'll help us identify it in the lab.
So it's a sponge stock? Or
yeah.
Sponge stock with an encrusting octocoral on it.
Yep.
Very cool.
Yeah.
Would it be of any interest to pick this one up or save that bio box for another?
Yeah, I would save the bio box. I mean,
okay. I'm just thinking it's
on a rock, right?
Well, in terms of time.
No, I mean, we have a piece of it.
Yeah, okay.
This is just a good observation. I just want to take a look.
Great.
Okay, full wide, please.
And, yeah, it's not like, that's a big rock.
Yeah.
That's not going anywhere.
We would not take that rock.
Well, you could take it, but you just won't be coming back.
You're right. This is true.
Anchor. Almost back to that where we were. Coming up. Pendulum swings. There we go.
Here we are.
Now this is the stuff.
Yeah, this is the stuff, and it's pretty extensive. So
Yeah.
I'm wondering if we might have some luck.
Well, maybe there's some fine-grained stuff underneath that'll provide a nice plug.
Yeah.
So you can get a decent core. Trick is getting through the top layer, I think.
Yeah, and we'll probably get a better view of how extensive this is once our disk swings around.
All right. Do you guys want to zoom in on this, or am I free to poke it?
No, you're free to poke it. I think we're good.
All right, Jeff, go ahead and push on in there a bit, please. Awesome.
It's an effective drill.
Oh, effective drill.
Oh.
Yeah, it's got some stickiness to it.
Yeah.
Yeah, that'll take a nice core, I think.
Do you want to push in on this dust cloud that I got going on here?
Like with the push core or?
Oh, just with the video.
Oh, I see what you're saying. Okay.
So we can see kind of, might work.
Yeah.
There's some stickiness to it.
Yeah.
Wonder how deep it'll be, but we can try it out. Okay. I'm convinced.
Kind of looks like what happens to your hands when you're making bread dough.
Go ahead and come wind up, please.
Yeah, there's quite a current down here.
Yeah. Just wash your hands.
Yeah.
You gotta wash them for at least 20 seconds.
Because there's a clean on deck.
All right, Jake, you want to switch over cameras there first?
Yes.
And you want to follow me on bubble too, once you get a chance. Thank you. Is it tricky to process a core sample, Beth?
Yeah. Especially when it's got a bunch of little rocks on top of it. So
So go ahead and bump it out.
a
like
bit.
there's multiple
Half
levels
of
of trickiness involved. Once you finally get a core
a little more.
and get it in the quiver. So at the bottom of those little gray tubes, there's a black rubber bung
There you go.
that
That's
sticks
good.
into the bottom.
All right.
And when the core comes back on deck, then you gotta get the core out of the quiver without losing the whole thing. So that's the first trick. And then you gotta transfer it onto a sectioning stand. That's your second trick. And then you gotta try to slide it down gently so that you don't disturb the sediment layers, so that you can then start sectioning from the top. So lots of places where that can go wrong, and you end up with sediment all over the floor instead of where you want it.
On a moving ship.
Yep.
Forget that part.
at like 3 a.m.
Yeah.
Sounds like fun.
This is not the best of grips. These are the worst of grips, actually, for this.
Snaggletooths.
Yeah, seems like with the coral cutters, you want the baseball holds, not the cylinders.
Or the hockey puck.
Or the hockey puck.
But a cylinder.
Anything but a vertical cylinder.
Are they all the same type of core here, or are they?
Yes, they are.
Okay.
All right. Come back.
Gives you an indication on that current.
Mm-hmm.
Could we get a zoom in on that before the push core goes in, please?
Sure. How about we do it up here then? Go ahead and push on in there, Jeff.
Thank you.
Like right about here. All right, go ahead and come a little wide there, please. That's great. Yeah. There. Might be as, we'll see.
Yeah, that might be refusal there.
Yeah, it might be as far as it goes.
Yeah. All right. Well, let's see what the future holds. Is it going to come out or not?
All right.
Ooh.
That is not a core. Want me to wash it out and try again? Looks like I lost all the plug.
Yeah, sure.
Jake, you want to look over on bubble there? Yeah, it all just slid out the very beginning. Currents. I blew it across.
Currents,
Okay.
yeah, there you go.
Nice. Okay. Sorry, Rose.
All right, we can wait it out.
All right, one more time. Do another close-up or?
Oh, no, that's okay. It's just kind of dealer's choice on where to try.
This guy's empty. It just has a little bit of leftover sediment. That's all right for you?
Yeah.
Okay. Sorry.